The last talk is by Vidya. Vidya is a PhD student in IIT Madras. She works with both Smita Srivastava and Neerav. So she'll be talking today about some of our computational work.
Good afternoon. So today I'm going to talk about the role of computational fluid dynamics in modeling and the scale-up of bioprocesses, and with a specific emphasis on plant cell cultures. So the overview will be like, first I'll be talking about the challenges in the modeling and specifically scale up of bioprocesses, and then how can computational modeling of fluid dynamics help in bioreactor design, and then I'll talk about some case studies where computational fluid dynamics has been used for bioreactor design, and then we'll look into the specific considerations in plant cell cultures, and then a short work of us where we have done a CFD based compartment modeling where we have predicted the biomass productivity of a plant cell culture via udarata, and then we'll talk about the opportunities, challenges and the conclusion. So coming to the challenges in modeling and scale up of bioprocesses, so mainly bioprocesses, they aim to maximize biomass or the product productivity. That is one of the main aspects in bioprocesses. So in that, the scale up is a crucial step, because when we go from the smaller scale to higher scale, there is a reduced yield and productivity, so that is going to hinder the process development. So for going from lab scale bioreactors to industrial bioreactors, there is a lot of velocity gradients and temperature gradients, which will lead to mixing issues and mass transfer issues. That is one aspect. And secondly, with respect to specific cell cultures like plant, mammalian and fungal cell cultures, depending on the cell characteristics, it is important to design the bioreactors, which is with respect to the impeller, sparger or the type of bioreactor in that case. And also with respect to the downstream processing applications, it is important to understand the different aspects of it so as to have a successful bioprocess design. So in all this, fluids, which is nothing but the gas and liquid, which is actually a heart of the bioprocess. So how can computational modeling of the fluid can help in having a better bioprocess design? So firstly, computational fluid dynamics is a technique where the fluid flow governing equations such as the mass, momentum and in some cases energy transport is solved numerically, because these equations are non-linear and coupled. So this numerical solving will help us to provide flow field information such as the velocity, the pressure and turbulence which is prevailing in the system. So this information can actually help us understand the biological system better when we actually look into computational modeling. So here we can see there are a lot of applications for CFD in biotechnology, right from fermentation to the downstream processing, but I'll be focusing only on the fermentation aspect, which is bioreactor design. So with respect to bioreactor design, we can actually get information like mixing time, the flow patterns and different bioreactor design aspects such as the impeller design, sparger design, and with respect to the operating parameters such as the impeller speed and so on. Apart from that, there are also aspects where coupling the kinetics of the cell with the fluid dynamics can actually help us investigate the effect of hydrodynamics on the mixing and mass transfer. And with that, there are different types of coupling with respect to unstructured model coupling, and then the next step is to have a structured model coupling, as we can see in this figure. And there are also certain cases where the particles, the cells are considered as particles and they are tracked along the reactor to see how their behavior is in with respect to the fluid. So these are some case studies where the first one is actually on the sparger type and the surface area of the sparger optimization in a 50-liter single-use bioreactor for mammalian cell cultures. And in the same study, they have also tried to optimize the impeller tip speed, that is the impeller speed for the optimum mixing. And then one more study where they have for a particular single-use bioreactor for mammalian cell cultures for biotherapeutics production, they have looked into mass transfer modeling, like whether they will be able to predict which particular design has a better mass transfer efficiency. Then coming to other aspects, there is another method where they have used a CFD based compartment model. So here what people do is like there is a CFD model which we are able to simulate. From that we can actually divide the system into compartments, and from those compartments we can say how the mixing, how the oxygen mass transfer and so on are prevailing. So in this case they have actually calculated how the mass transfer coefficient and the oxygen saturation is present in each compartment. This is actually a 50-liter bioreactor, so it is useful to look at such systems when we are working in higher ranges. And there is another study where they have coupled kinetics here. So in this study only the substrate utilization and the oxygen mass transfer was coupled, but in this study they have coupled both the biomass kinetics and the substrate utilization kinetics and they looked in a 5-liter bioreactor and in a 50-liter bioreactor. So within a time period of like 120 hours they were able to predict how the biomass grows and how the substrates are being consumed in the bioreactor. So they were able to compare between a 5-liter bioreactor and a 50-liter bioreactor. So with all this background, looking into plant cell cultures in specific. So plant cell cultures are basically it's a technology where we grow the plant cells in suspension. Why? Because for the production of products, if we go for the normal extraction from actual natural plants, there are a lot of inconsistency in the product yield and also the non-availability. So in order to produce high quality plant products, we go for an in vitro cultivation of plant cells in suspension. But the problem in plant cells is not with respect to the next step of bioreactor scale up, but actually when we go from shake flask to the reactor itself, there is a dip in the biomass and product productivity. So we are looking into this question on how we can use computational fluid dynamics to address this problem. So there are a lot of specific characteristics for plant cell culture. Like first of all, the plant cell cultures are large in size and they have aggregate formation capabilities. They are shear sensitive, and since they have large size, they have actually possibility to adhere to the bioreactor parts, and there is a problem of insufficient mixing and settling of the cells, and there is because of all this there is an insufficient oxygen mass transfer, and there is also a non-Newtonian rheology. So specifically with respect to shear sensitivity, CFD has been used in 2 different studies. One in 2001 where they have looked into the cell culture's cell viability when it is grown in a stir tank reactor, but they have not done for the entire reactor, but they have just taken a annular section which actually mimics the impeller turbulent region, and they have checked the velocity profile at a high RPM and the energy dissipation rates, and they also looked into, with respect to this energy dissipation, how the viability is changing. And in another, this is with respect to carrot cell culture. In safflower cell culture in 2016, they have actually modeled the stir tank reactor, only this particular region, like just for taking it as a computational domain, and they have looked how the shear stress is being distributed in this region and how the shear stress is related to the death rate of the culture. So what we are interested in looking is looking into this problem of how we can take the shake flask comparable biomass productivity in reactors. So one particular problem is that when we look into the bioreactor design, we actually think of using different modified impellers. So once we see that the biomass productivity is dipped in the bioreactor, the immediate thing to change is to see how whether we can change the different impeller sizes or whether we can change the reactor configuration itself. So this is actually mostly a hit and trial process, and that is going to take a lot of time because plant cell cultures take a longer duration to grow, like up to weeks to months, and also the selection will depend on the species. It is not a common thing which can be useful for all the cultures, because some cultures have a very good suspension ability, some cultures have lot of large aggregates. So it is again becoming a very species specific thing. So if we are going to do it in a hit and trial manner, it is going to take a lot of time. So whether we can look for a rational approach to do this. So we have thought of looking into using CFD to actually see whether we can see the effect of hydrodynamics on the biological reactions by coupling the hydrodynamics or the CFD data with the kinetic model. So the first step is to develop a CFD model which can characterize the fluid flow in a stir tank bioreactor, so for a particular hydrodynamic condition, and then whether we can integrate this model with a pre-established kinetic model which is accounting for a biomass and substrate utilization. So for the first step we developed the geometry of a 3-liter bioreactor which we have in our lab, and we divided the entire domain into like multiple regions which is called as a meshing. So and the temperature we have chosen will be the two phases which we are choosing will be the air and the plant cell suspension at the temperature 26.6 degrees Celsius. This is because the plant which we are interested in, as I said earlier, Viola odorata, as a model system we have taken that plant, so that grows well in this particular temperature, and we actually also modeled shear that may exist in the flow field as a function of the cell aggregate diameter. So we used ANSYS fluent software to solve the governing equations of fluid flow in this condition, where we used a Eulerian model because this is a multi-phase simulation where we have both air and water, and we used a multiple reference frame approach for the moving domain impeller, which is like, so we have taken a particular section here around the impeller which will not be moving actually, but the we actually assume that the fluid around it has a relative velocity such that the moving domain will have the velocity as that of the impeller, but it will not be the actual mesh motion, and we used a standard K epsilon model for modeling the turbulence phenomena. So we were able to get a velocity pattern which was in the which was actually had a good axial mixing. So the impeller which we have used is a Cetric impeller which has been known to be good for plant cell cultures for a particular plant. It has been published earlier. So what we did was we used that impeller and a porous sparger. So for that we were able to see a good axial mixing, and we also saw that the theoretical tip speed matched well with the CFD predicted tip speed, and we also did a grid convergence study where we were sure that whatever meshing we have done was, whatever results we are getting is independent of the meshing we have done. So based on that, from the velocity profile which we plotted with respect to the bioreactor height, we divided this reactor into two compartments. So this will be the compartment 1 and the compartment 2. So we divided the compartment based on the velocity data. So this is an initial approach where we have used only two compartments. So when we divided it, and then what we did was we assumed that each compartment has a uniform mixing. So based on that, we took a kinetic model which was established in our lab for Viola odorata cell cultures, and we actually calculated the flow rates between which flow of exchange between these two compartments, assuming there is uniform mixing happening within the compartment. So we coupled these two equations and we solved the biomass production kinetics and the substrate utilization kinetics using a Python model. And we could see that the compartment wise we were able to see the evolution of biomass, the substrate sucrose and potassium nitrate. Why these two substrates were? Because these were the limiting substrates for this particular cell culture, and we simulated for up to 12 days, and we were able to see that it actually the CFD predicted model actually matched with the evolution of biomass and sucrose from that of the experiment. And from this, based on volume average value, we calculated what will be the biomass productivity, and it was around 1.7 gram per liter per day, which is quite close to the experimental data. So now we are looking into improving this model to actually see how the biomass and the substrates are with respect to more specific zones rather than two compartments, looking into more than two compartments. So we also can actually use this prediction of biomass productivity. We were able to establish that this CFD model, when linking with the biomass kinetics equation, we are actually able to predict for this hydrodynamic condition what will be the biomass productivity predicted. So we can actually do the same process for different bioreactor designs in silico and see which has the maximum biomass productivity, and also we can look into different bioreactor configurations and see how whether some other bioreactor configuration is going to help the system grow better. And also we can account for the non-Newtonian nature of the fluid. Right now we are not looking into that. So if we can see whether the non-Newtonian nature of the fluid is going to have an effect on the plant cell kinetics, and also we can look into Euler-Lagrange particle tracking. That is, we can take each cell as a single particle and see how it is moving around, and whether we can account for cell settling and cell adhesion also, so that we can look into these problems which are common in a plant cell culture bioreactor cultivation. So in conclusion, the challenges in scale-up and bioreactor design for cell cultures are paramount, and CFD has actually been a boon to study the effect of fluid dynamics on cell physiology. So with respect to plant cell cultures moving from shake flask to bioreactors, generally hit and trial methods are being taken to modify the conventional bioreactors. So reproducing shake flask biomass productivity in the bioreactors is a key problem right now. So we have looked into a rational approach where the CFD based compartment model kinetics, which actually can relate the interactions of hydrodynamics and or the effect of hydrodynamics on biological reactions. So this proof of concept, whether for this given design and operating conditions, we were able to predict the biomass productivity. So this can be replicated for an optimization of which is the best design for biomass productivity for any culture, if we know the biomass kinetics. So I'd like to acknowledge my supervisors, Professor Smita and Professor Neera, and Professor Kameshwar Rao for collaborating with us and helping us. And I also thank our research group, Ministry of Education, for funding, and thank you.
Thanks, Vidya, for this nice talk and also staying on time. So we have time for a few questions.
Can you use your Euler-Lagrange particle tracking of cells to help with dealing with the problem of biofouling in ships? Thanks.
Yeah, I'm not sure about biofouling concepts, but I think like you mean like settling of cells and all that. Yeah, but actually Euler-Lagrange particle tracking is something where we track the cells just by looking at the adhesion surfaces and how the movement happens. Maybe we can do that, but I'm not very sure of it because I'm not aware of the idea.
Hi, very nicely presented. I have a couple of clarification questions. So the growth model you had, if you can go to that slide. So which variable there was related to the prediction of the CFD?
So this is actually the specific growth rate, but the way we linked it is by actually using a mass transport model, mass transport equation for the biomass and the substrate. So in that, the rate of reaction was this mu.
So substrate is the carbon source?
Yes.
Okay. So essentially the CFD is predicting the distribution of sucrose in the reactor. Is that?
CFD, we use CFD to look into the velocity patterns. So
Sure.
that velocity pattern in steady state, we use those data in order to calculate the exchange between the two compartments. So we use that to actually write a mass transport equation for biomass and sucrose for every compartment.
Right, I know. But so my question is that, let us say you do not carry out this complicated analysis. You say it is an ideal CSTR, right? What will differ? I mean,
okay, in an ideal CSTR, we'll be only solving the kinetic equation and we will get a biomass productivity. But the main idea of our study is, if suppose tomorrow I'm going to optimize for a different design, so that particular design will have a different hydrodynamics and velocity patterns. So whether I can predict for that, because doing it experimentally is going to take a lot.
Sorry, I'm just unable to. So hydrodynamics will give as the output a velocity profile, right? But if your growth is not dependent on the velocity profile, then how does it matter?
Okay, so right now we are looking into the mixing, like whether the between the compartments how the movement is.
Okay, fair enough. But for mixing, again, if it is a small molecule that is being mixed, then beyond a certain velocity, it is only a matter of whether it is microseconds or milliseconds, right? So will that, compared to the growth rate time scale, it may not matter in the sense. So
yeah, I understand your question.
sorry, and a follow-up suggestion
Yeah.
is that what actually might matter is in dead zones there may be clumping. And have you thought about that?
Yeah,
That's
yeah,
my
we
last question.
Yes, thank you for the suggestion. I'll look into it.